you. I want to move on now to clinical outcome assessment tools and digital health technologies. Nimi, I'd like to start with you. Talk to us about your thoughts on like I mentioned, the COA tools or clinical outcome assessment tools and the DHTs, digital health technologies, to support cell and gene therapy product development.
Hi, Erin. Thanks for the question. Very fast-moving field on the use of clinical outcome assessment tools as well as digital health technology tools or digital health technologies. Super important for any product development, especially for cell and gene therapy products in particular, because some of these products are administered and after that you're collecting patient data, which ideally could happen in the remote setting, and the patient does not need to come to the clinical site unless they're absolutely needed to be there for certain kinds of data collection. And that's one point I would make why this is more important for cell and gene therapy products. The other point I would note and highlight is, while we do understand that some endpoints cannot be adequately replaced solely with clinical outcome assessment or digital health technology data, there might be worth considering identifying those circumstances in which these data, that is COA and DHT data, could serve as a sole source of safety or efficacy data. And some things to consider when thinking on using COA and DHT is to collect patient-level data that would be most useful for drawing conclusions or informing regulatory decision-making, and to proactively deliver the data collected for study requirements to the sponsor, thinking of those data quality related requirements when trying to use COA and DHTs. And really partnering with FDA, what we see is a lot of the COA data in the form of patient experience data or data using digital health technologies or wearables and things like that, gets submitted to the FDA, but may not always end up in the label or in the table that FDA populates or truly informed regulatory decision making. And it does take resources on all ends and adds to the cost of drug development to incorporate COAs and DHTs. I think partnering more closely with the agency and thinking about ways to ensure that the data collected and the resources invested do end up in informing regulatory decision-making and the safety and efficacy profile of the product. But that is the future. And lastly, as you can imagine, there are learnings from the COVID pandemic and how especially digital health technologies were incorporated during the COVID to continue clinical trials when sites were closing, or to continue to collect data in remote setting for patients. And so it would be helpful for all of us as an industry in partnering with FDA to share and apply learnings on flexibility on use of DHTs obtained from the decreased ability to conduct in-person data collection during the COVID-19 pandemic.
Wonderful. Thanks, Nimi. I want to move on to post-approval monitoring. And Bob, I'm hoping you can help us out with this one. So the FDA recently held a workshop on post-approval safety and efficacy data collection for cell and gene therapy products. What are some of the key considerations for post-approval monitoring, safety, efficacy data collection, and then the long-term follow-up for these cell and gene therapy products?
Yes, and I think the registries and the follow-up is very important to determine the long-term effects. And I think in some of the cases, it's always the question is how much data are necessary for approval and how much additional information can be collected and gathered in follow-up. So prior to approval, is it 3 months, 6 months, or a year of follow-up, or longer versus having the approval earlier on and then following those patients in a follow-up protocol, either registry or a long-term follow-up study. I think those are important features. Then for cell and gene therapies, it's the requirement for a 15-year follow-up. That is not as intensive or extensive, but it could be checking what were the medical conditions you encountered over the year's time. Were there any malignancies? Were there any problems with end organ damage that were encountered in those individuals over time, to collect that information, to follow up to see what are the long-term consequences. And I think over time, as we gather more information, we'll have more confidence in the safety profile of these particular products to continue them and use them more routinely and cut down on the amount of monitoring as the case may be. I think in some of the cases, such as for the recently approved product where there were 3 reported cases of malignancy in children, obviously that's going to be looked at very carefully. And there are programs in the post-approval monitoring program where you look at if anybody develops a malignancy, how do you determine whether or not it could be related to your therapy? And there are specific tests, genetic testing, as well as deep sequencing to determine the potential risk of the product to cause that malignancy, or to totally rule it out. And we're in the infancy with that type of monitoring, but more and more as the scientific knowledge increases, we'll be able to predict that and determine whether or not it is actually due to the cell or gene therapies that are being prescribed.